So I collected all these urine samples and took them to New Zealand. Okay, and that was even funny. At the airport, they stopped me and said, "What is this?" They took me to in a special room and all that. I said, "Look, this is just frog urine. You can do whatever you like with it because in the tubes itself, it is colorless." And I can see my supervisor was at the airport and he hid him, and maybe Edward has brought something with him.
He's just there sitting there waiting, going, "Oh, it's got to be that froggy urine."
I can't think of any more human activity than conducting science experiments.
The game I play is a very interesting one. It's imagination in a tight straitjacket.
The beauty of a living thing is not the atoms that go into it, but the way those atoms are put together.
What I always think should be the basis of education is not answers, but questions. We should teach kids how to question.
Yes, thanks for this. I am Dr. Edward Narayan. I am a senior lecturer in animal science at Western Sydney University. My core research is in comparative physiology, and we apply physiological techniques to understand how animals respond to their environment, basically.
Well, before we started this podcast, we were talking about how you began your journey into this field. Talk to us how this all started. How did you get fascinated with science and particularly in this field?
Yes, it's been a long journey, as if it's been over a decade now since I finished my PhD. And the journey started quite early. I grew up on a farm with my parents, and Dad used to keep cattle, and Mom kept chicken and poultry. And growing around animals in a village back in Fiji was really worthwhile for me to have a connection with nature. And so coming from a education-driven family, we were always keen on getting high grades in the class, and getting the scholarships that we need to get ahead of the group. So it was pretty much what we had got on the plate, and being able to harness or make the most of education and the opportunities that we get, especially coming from remote rural areas. So I studied animal science as an undergraduate student back in Fiji, and that was looking at biology of animals, and got trained in physiology as well. And then during that time, there was a bit of an interest in conservation biology, because scientists around the world were recognizing that animals are facing a lot of endangerment through the disturbance that humans are causing to the planet right now. You can see, forests being cut down. Climate change is a big issue, and when that happens, animals start to suffer and they, become sad. So that drive was always within me, how I can sort of contribute something to science. It is not always written in gold for anybody. Career actually has its own, it's a destiny. So a lot of luck was involved for me, especially at that time when I was doing my master's. My professors were very switched on. They were quite enthusiastic about conservation of animals. And so at that time, there was this frog, virtually nothing was known about its biology. And so they said to me, Edward, we'll give you a scholarship. You've done undergraduate, you got a gold medal in biology. We think you can do this. So for 3 years, I was basically in a small island studying the biology of this frog, of which virtually nothing was known. So that was a big exposure for me, getting to work with the local people. knowing the local culture and the Fijian culture, and understanding that. So
this was for your master's, not for your PhD.
this was
right?
for my master's. So So basically, I started my master's. I did my undergraduate for 3 years, and within that, I got prizes and all those things. And then I started a master's. That went for a year. I came to Australia and gave a talk at the Society for Conservation Biology meeting here in Sydney. And I won a little prize or a certificate as the best student presenter from Pacific Islands. Amongst a crowd of 1,000 people, I was called upon, and I can still see myself going down it was a big lecture theatre at the University of New South Wales at that time. It was a big exposure for me, to stand amongst elite, scholars here in Australia. That was something that really gave me oomph,
confidence, I bet.
confidence. Yeah. So then I went back to Fiji during my master's itself, which is a postgraduate degree that I was doing at the time. Going back and forth, coming to Australia and New Zealand, I think I traveled around 10 times coming out in Fiji. And it was a funny thing, people thought that I had migrated.
Because you were here so often.
And I was here so often, and I said, whoever is upstairs, I said, one day please send me for good.
Yeah.
And, destiny was there. And at that time, not even I had submitted my master's, because I had already published papers in international journals within my master's.
Right.
There was an opportunity at that time for a PhD scholarship. So I took it as a gamble. I sort of took my CV and my gold medal that I had and my publications to the interview. And my dean at the time, he was from the UK, and he was quite visionary, and he said, Edward, if you can do this, we will give you an opportunity. So you'll basically jump straight from master's and do a PhD now.
Yeah, that's awesome.
So it was something, people were realizing and recognizing the effort that I was doing as a student. I was listening to my professors. I was being able to apply my undergraduate skills. Because in undergraduate itself, our students pick up a lot of hands-on skills. And that goes a long way when you are actually doing research, whether it's master's or PhD. So I said to the interview panel at that time, look, give me another 2 years. I will do, more research and be able to convert this into a PhD. The thesis,
show us your PhD.
the
You're showing
thesis
us
which
before.
is basically over here.
Yeah.
It's legit.
Yeah.
When was
So-
that? When did you get? Is that 2000 and what?
9. Yeah.
2009. Wow, time flies.
That's a thick one. Yeah.
Yeah, so I had to get rid of a few chapters, otherwise it would be a bit thicker. Oops.
Just be careful of that. So tell us about your PhD and master's research, I suppose,
Yes.
because you combine those two. So you're looking up this frog. Why?
Because sorry, just to interject. Your PhD was on the same frog, wasn't it?
Yes, so I was just trying to explain that whole journey. So basically, the master's was on the captive breeding of the frog. So to be able to do that, I was actually gonna bring frogs from this island back to the university. So I set up a captive breeding facility, and no one had ever seen eggs of frogs of this particular species.
Right.
So naturalists have been visiting Fiji for many decades now, and there was anecdotal evidence that the eggs look like this.
Just talking to the mic if you want. Sorry.
So naturalists have been visiting the Pacific Islands for so many decades, and so they were observing this beautiful creature on this island, which was having unique breeding characteristics because they don't have tadpoles. So when you look you think about a frog,
Yeah.
people think that cane toad is a frog. We've got cane toads here, and we've got them in Fiji as well. Cane toads actually have got a tadpole, so they have got a tail. The baby actually swims in the water. This guy actually lays egg like a chicken.
What?
Yes, and the baby basically develops in the egg itself. So this is known as a direct developing species. And that is one of the reasons why these particular species are quite prone to environmental threats because they have a very short breeding space, and they lay only less than 100 eggs compared to a cane toad which lays 1,000 or 10,000 eggs at a time. So they entirely procreate or reproduce entirely on land, and so that was really a big aspect of my doctorate, which was to try to initiate their breeding. So it was a lot of hard work, setting up the breeding chambers and getting cameras like this so that we can monitor the activities remotely. These guys are pretty shy. They don't want to, demonstrate their mating rituals out in the open. So you are, still, it's like needle in a haystack kind of a situation. So I had to.
I guess you have to set up the enclosure, so it's all right. Just move over, Hamid. I guess it's very difficult with a lot of species like that as well, because you have to set up their enclosure in a particular way to encourage them to breed.
Yes,
and if you hadn't seen them in the wild breeding, then you may not know what those conditions are.
Definitely, I think you've raised a very interesting point there, because the breeding biology or the breeding ecology of each species is unique. And being a terrestrial frog species, it had really unique requirements in terms of the substrates it needs, the temperature, the different refugia. So basically I brought a little piece of the island, its home, on a boat back to the main island.
Amazing.
Yeah, and at that time I was quite lucky because there was a guy from Australia who was visiting Fiji at that time. He became my best mate and he helped me quite a lot in setting up the enclosures. So, there was a helping hand. Like I said, a lot of luck was involved in my journey, and that was really useful. And so within the masters itself, nobody had ever seen the frog's egg. I was working hard, blood and sweat, recording data every day. I was in this cage. It was basically visible to everyone. People going past me were saying, "Oh, who is this guy? What is he like? So dirty and everything." And the other students were looking at me. What is he doing every day in there? It was part and parcel of the fun being a biology student. And so
it sounds like a lot more fun than what we do, isn't it? We just play with cells all day. It's actually quite boring.
Yeah, you have to.
I think the fun is that we don't have to try so hard.
And that was the thing, because I was running against time as well. So it was the Festival of Lights back in Fiji at that time, and I don't know if you know, it's a traditional Hindu.
Is this Diwali?
Yeah, Diwali,
Yeah,
and
it's a
during
very good
Diwali, my frogs laid eggs.
Oh, wow.
On the day of Diwali, I went to the cage this morning feeling down, and I've been working hard for over 9 months now, and I went to the cage, just a routine husbandry, a routine check, and I was cleaning the soil, and I saw marbles. And with little tiny something moving in it. At first I thought it was earthworm. And then I said, "No, Edward, look at this. This is the ground frog eggs. First time ever. Has anyone seen that?" Yeah. I ran into my supervisor's office. You can imagine, I'm still feeling that now. I can see myself doing that. And it was like climbing 100 steps up into their offices. And I said, "They have bread. They have bread." And, 10 minutes later, news cameras, everything came. The national TV was there. Yeah, and my supervisors, it was funny because one of them is a conservation biologist and the other is a physiologist. So they were starting to debate as to what should we do with these eggs. Yeah. And I was in the middle, sandwiched amongst them. I said, wait, guys, I am the student here. I need to, wrap up this. And because the physiologist said, let's
break up.
The
conservation biology.
And let's conserve this. And that's entirely true, Hamid. The physiologist was saying, let's put one under the microscope and see, you might discover something totally unique. Right. And the conservation biologist said, no, let's take, 10 of them and release them on this island and see what happens. So, and I was in the middle as a student, I was just 22, 23 at that time, and it was like, Edward, what should I do now?
Yeah, that's amazing. So you were just a master's student.
Yes.
All good. You were just a master's student at that time. Like, what is that like? having to, you said, confront the press immediately after you found it, and then having to deal with two different supervisors who have conflicting kind of. What is that like?
That's entirely true. It's part and parcel of the game, I guess, or whatever you sign up for. But the good thing was that the supervisors were understanding, of my requirements and what the requirement of the degree was. So, putting passion aside and putting debates aside, we came to a consensus that no, let's look at the developmental biology of the frogs. The media was there; they were quite good. Similar to you guys, they helped me in taking some cool pictures and putting out a little news article on the national news that evening. It came out. And all of a sudden I had to be on the news. So the whole of the island, the whole of Fiji basically saw me on the news, and that was a big thing because we had bred the frog successfully for the first time. Not that nobody was trying that before. There were several zoos in Fiji, a few, not many, like not in Australia. There was one zoo that was trying to breed them. The funny thing, they bred the frog, but it wasn't a frog, it was a lizard. Yes, so nobody had done any research on the actual biology of the animal, so nobody actually knew how the eggs looked like. So, and sometimes even the snail eggs look pretty similar to a frog's egg. So they are shelled, and
they found the eggs, but when they got them back and bred them, they were actually lizards.
They were actually lizards,
Or snails.
and not a bad thing because that lizard is also endangered under the IUCN criteria. So you've saved a species by accident.
Yeah.
So tell us, what were the key, I suppose, ingredients that led you to breed them successfully versus the zoo?
Yes, and that is a very, very interesting question. The main thing is give them time and space. Let them do their thing. Don't disturb them, because what the zoo was doing, they were trying to watch them every day, virtually not using a camera. So I was using a remote camera, like this is the breeding chamber. I wasn't there. I was observing them using a camera, and whenever the breeding period was about to start, I stopped going there.
Right.
So during the Festival of Light period, that's when the rainy season starts in Fiji, and the frogs actually cue themselves to the rainy season. And so I stopped going there quite often, and I think that gave them an opportunity to do their thing.
Right.
So yeah, it is very important not to disturb animals, especially if you are trying to breed them, because what happens, these females, they have eggs in their underbelly, and if you harass them quite often, they will actually release the eggs without fertilization.
Right.
So that is very important if you're trying to breed a species in captivity. Yeah.
So prior to your research, did people know that they were terrestrial breeders, or what did they mainly think that they would actually have quote unquote eggs and that would turn into tadpoles in water?
Not the naturalists, the ecologists. We knew that there was like I said, an anecdotal evidence that this is a this is a terrestrial breeder. Okay. But to any person down the road, any man on the ground, a villager, they wouldn't know that they had nobody, like a person on the ground who is not a scientist or a researcher, they didn't know that. So not that the science world was not totally naive of it. They were aware that yes, they do, because there was naturalists who had been to the island and seen some things. They made a note.
Yeah.
But there wasn't a real science sort of experiment as such to be able to demonstrate this, because once the egg then bred and the two supervisors agreed that yep, Edward should observe them under the microscope, I actually studied the whole developmental biology. So from the time the eggs were laid through to the hatching itself, and I recorded it in it's actually like a ethogram. It's like a birth chart of a frog. So I made this birth chart for this particular species, and that was published in the Journal of Zoology in London, which is a very prestigious journal. It's high impact, and still I'm within my master's, being able to do that, which was a big tick. When I went to this PhD scholarship committee, they said, Edward, you have done amazing, So that was really important for me to make sure I keep writing. I keep talking to my supervisors as a student and publish. And that was really important. Yeah.
So how would you study what's happening inside the egg?
Yeah, so it's basically non-invasive. So you have to observe it under a microscope. There's a special microscope. You put the egg with some water in it and let it float, and then you have a microscope similar to this mic and just observe it so that it enlarges. And you don't take all of the eggs, okay, and just a subset of it and observe it. And then see how the actual...
so you actually have the shell still on it?
No, so the frog...
You crack the egg in the water and you observe what happens.
I can see you are getting very excited with this question. So this is not a bird's egg like with the shell. The frog's egg don't have a shell. They are shell-less.
Okay.
So you
can see through them.
You can see through them. In the beginning, they will look like just the moon. Let's say maybe I can show you a picture or two.
Yeah.
Is it kind of like a human egg, right? Because humans
Yes.
have oocytes.
Yes.
And
Yes.
embryos hatch and they're all transparent.
Yes, and you can see here.
I'll pass it to you and you can hold it up to the camera.
So that's actually a bunch of eggs.
Can I pass it to
Yes,
you
please.
can hold it up to the camera.
Okay, let me just look at it. Okay. Yeah, do you want to describe what's happening?
So that's actually a freshly laid bunch, or you can call it a clutch of frog eggs, which I have basically cleaned. They look quite shiny. And what you can see there is the actual oocyte or the fertilized egg. If you put that under a microscope, then you will basically see the germline, which is the embryo sitting on the oocyte plane, and that will take 30 days to develop into a mature embryo, which we can show here as well. If I can go back to the page. And you can see, maybe Hamid can turn the pages as we go along on the camera. And as development progresses,
This is a slideshow of his thesis.
Yep. Is it clear there? So
No, a bit closer.
As development progresses, you can see you can see the head and the whole vertebrae. And basically the second slide, you can see the embryo sitting on the oocyte. So he's getting air, nutrition, and everything it needs to be able to grow. If you turn the page, maybe you can see more. So yeah, you can even see the vertebrate column. The
VC.
Yeah, that's it. You need to label all your diagrams so that it's clear enough. It basically takes around 30 days for it to grow into a adult frog like structure.
One question: human beings, the embryo gets its nutrition from the mother in the womb. What about the eggs of frogs?
The ovum, the oocyte itself.
So it's got
everything, all the ingredients it needs.
Just like the
chicken
white.
Egg.
Yeah.
The egg
and
white,
human eggs
yes,
as well
yes.
have a similar thing, I think.
Do they have?
Yeah. It's because we all came from a common species.
I thought evolution was fake news.
and at the end, this is what you have. Okay, it's like a little froglet.
That's cool.
So that's a freshly laid froglet. It's just being hatched from the egg. And the way they hatch is also very interesting. So what happens? It's the frog has got an egg tooth, so it uses the egg tooth to actually to break into the actual gelatinous layer, and slowly, and we actually observed that happening. I've actually got some video as well, and they jump out of it. And for the first 5 days, the froglets, now we can call them a froglet. So as you can see, the development actually does not have a tadpole stage, so it's all inside an egg. And as it actually gets into a froglet, it reabsorbs its tail.
Yeah.
So all the fat in the tail is also nutrition. Okay. So for the first 5 days, when the froglet is actually on the ground, it still is absorbing some of the egg that it has in its underbelly. So for the first 5 days, it does not feed at all. After 5 days, then it starts to feed. So if you look at my journey, basically, if I can wrap that bit up. So what happened was the frogs had bred, and then I had published, and so that was like half of a PhD.
Yeah.
Okay, then what is the other half? At that time in New Zealand, there was a similar endangered species that was heavily declined and all that. It's called the Archie's frog. Which is also direct developing. It's a very unique lineage of frogs when you look at the history of the frogs itself. And at that time, they were trying to actually come up with non-invasive methods to measure hormones. So we haven't talked anything about the hormone biology.
Yeah, that's what I was going to suggest, that we probably get to the research that you're doing now, because now you're looking specifically at the stress response that amphibians have, and as you just mentioned, trying to analyze that by looking at hormones. So when humans get stressed, we have release of hormones through our bodies. And I'm guessing amphibians are the same in that respect. So we can probably infer things about their level of stress in regard to by measuring their hormones. So how do you go about that?
That's basically the second half of my journey. Basically, at that time, I had done the captive breeding. My supervisor, who was the conservation biologist, said, Edward, we have a collaboration in New Zealand where we are trying to come up with non-invasive methods of measuring hormones in animals, and in this case, frogs. And he said, have you seen a scat or a poop of a frog before? I said, yeah, I have. He said, can you touch it? These are some questions that supervisors ask, because for the next 4 years or so, I was just collecting frog urine on the same island where I brought these frogs from. So the reason why we are using urine in this case is it was quite accessible. And you asked the question of stress. Hormones regulate a lot of our biology. Okay, so whatever we are perceiving in our environment, it is all regulated by hormones. These are biomolecules inside our body that actually help us to respond to the environmental change. Okay, so if there is a change in temperature, there will be some hormonal changes. Or if there's a threat or some predation or something, an animal will need to escape, there will be a production of adrenaline, let's say the knee-jerk response hormone. So we can't always collect blood from animals because that is a stressful event itself. So at that time, this theme of conservation physiology was emerging. Conservation physiology, which is basically applying physiology into conservation biology. As you know, conservation biology is all about saving animals. Okay, we are trying to apply science to recover species from the extinction risk. Because many of our species are going towards that extinction risk. They might get extinct in the next, 50 years or 100 years or so. So my PhD supervisor at that time, Craig Morley, he was from New Zealand, and he had friends in New Zealand who were trying to come up with this technique to measure hormones non-invasively in frogs. And he said, let's start doing this with your frog as well. So there was one of my friends who was studying the Archie's frog in New Zealand, a girl actually doing a PhD in New Zealand, and I was doing a PhD in Fiji. And so there was a race as to who finishes first. So who publishes first? I published first, and I acknowledged her. We became, we're still friends. That's, I think, also important, is to maintaining a network with your colleagues and have a friendship sort of with the people that you work with. It's a healthy competition in that way, that at the end, so I collected all these urine samples and took them to New Zealand. Okay, and that was even funny. At the airport, they stopped me and said, what is this? They took me to in a special room and all that. I said, look, this is just frog urine. You can do whatever you like with it, because in the tubes itself, it is colorless. And I can see my supervisor was at the airport and he hid him, and maybe Edward has brought something with him.
He's just there sitting there waiting, going, it's got to be that froggy urine.
So I had this big styrofoam box. It's just the journey I'm trying to tell you. It's a big styrofoam box with over 2,000 frog urine samples.
Yeah.
I was collecting frog urine for over more than a year now on this island. Going from home, I live on one of the towns in Fiji. When I was there, taking the cab to the bay, basically where the boat will pick me up, and then take the boat, go to the island overnight, collect urine, frog urine, and then I was doing that on a monthly basis, not even having any experience on whether there will be any hormones in the urine. Like even today, we've been collecting crocodile scats to measure stress in farmed saltwater crocodiles, and the student asked me, said, "Edward, is there going to be any hormone in this scat?" I said, "Mate, I asked the same question, more than 10 years ago."
So you didn't try to do like a pilot study to determine if what you were collecting was actually going to work?
That's a very good question, Hamid. But the problem is I am in Fiji with very limited scientific resources. Okay. So I need to just trust the experts at that time. And then I think that is a very important thing that student needs to realize, that experts have a lot of experience. And whatever they say, 99.99% of the time, it will be for the benefit of the project and for the benefit of the student. So I trusted my professors. That is very important as part of a student's journey, to have trust, and not blindly. I was aware of the frog physiology, so I knew that at the end, once the hormones are metabolized, they should appear in the excreta. That is fine. As long as my sampling technique is good, I should get something out of it anyway.
Right.
And at that time, it was not me alone. They were sending me updates on the Archie's project. So they were sort of starting to measure things already by the time I even took the plane. So anyway, that journey at the lab in New Zealand, this was at the Land Care Research Facility. That itself was interesting because I had only 3 months to finish my PhD. So, sitting in the lab, getting to know the technique on the spot. I can discuss some of the experiences that I had there as well, if you like.
I was interested in so obviously, like, developing new techniques in science is incredibly important. Like, a lot of research just hinges on developing that assay.
Yes.
There was a recent study, really recently, just released, I think in part with the Garvan Institute, where they found a different structure of DNA in the nucleus. In life. You saw that?
Yes.
But the way they did that, right, was the interesting part wasn't finding it. Well, that was the interesting part, finding it in the live cell. But that wasn't the difficult part. The difficult part was designing the antibody. So,
very good.
So they designed the antibody that can find the stuff.
Amazing.
And then after that, it's just.
I think you should be
you
should become my PhD student. Stop doing this. You are quite.
So I guess my question is. And here's where you might not want me to be a PhD student, is then how do you do that, right? What steps do you take when okay, you've got the urine, you want to develop the assay?
Definitely, yeah.
What's the next step?
Yeah, and that's an amazing point of view. I hope all my students could say the same thing. The thing is, it's part of the validation. So you are now trying to develop this assay that will, let's say, for example, the tree kangaroo or the red panda. Okay, so somebody comes to me and says, "Edward, I'd like to study the red panda in Nepal and collect its scat samples, and I know there's a forest being decimated, and I want to show the world that the red panda is stressed." So you've got a very impactful research question. My job as the expert in endocrinology or animal physiology is to be able to come up with an experiment that can help answer that question or the hypothesis. It is very important to actually validate your assays. And developing that antibody is part and parcel of it. Because if I had used a rat or a human antibody for my frog research, it might not have worked. So what we actually did was, it's part of the validation whereby you spike the animal's stress response by stimulating its endocrine system with a precursor hormone, So we did that with a subset of frogs in captivity with the facility that I had in the university. It's actually called ACTH. So this is produced by the pituitary gland, that is like a messenger going to the adrenal gland where the actual cortisol is released. So we purchased ACTH and injected it into the frog humanely, and basically collected urine samples before, during, and after the actual stress activation. And then that sample was my big tick in the box for the biological validation. We did see big spikes in stress response in urine after the physiological validation. So that is a big tick, because that has now answered that yes, your assay is working. You are actually picking up physiologically relevant responses in the urine sample.
Right. Just a question. I mean, how different is the cortisol in amphibians versus other animals or ourselves? Weren't there immunoassays that could detect cortisol already?
Yes, so the cortisol is quite highly conservative. These are actually called glucocorticoids. But in frogs, it is called corticosterone. Okay, so it is slightly different to the cortisol that we humans or elephants produce.
Right.
So it is possibly the side chains are different.
Okay.
So the antibody that's available on the shelf for humans may not work for frogs.
Yeah, because maybe we should just fill in kind of how an immunoassay works, because a few of our listeners might not actually know what an immunoassay is yet.
Yep.
So maybe would you be able to do that for us?
Yeah, definitely. It's actually something that we are doing right now in one of the units that I'm teaching. So I teach a third year unit in animal science called animal production. And within that, we actually came up with a set of practicals that gives students hands-on training in analytical skills. So pipetting, how do you pipette? Okay, how do you evaluate a volume of something? It is very important that we have, like what I say to my students is, you are developing a toolbox. At the end, like we say, we are preparing, Western Sydney University is preparing students to be job ready. It's not just all about going and grooming a cattle or feeding a giraffe at the zoo. You may end up in a lab. And I have had that experience of being in the field and being in the lab. So it is very important that you understand how these techniques work. So the lowest volume that I work with is 1 microliter. Okay, which is like 1,000th of a milliliter. So 1 microliter, and I'm doing research on looking at welfare of sheep. We are looking at Asiatic black bears, the impact of bile farming on bears. At the moment, we've got a big project on koalas, looking at their response to noise and habitat destruction. Some really huge project on big cats, looking at stress with response to poaching in India, and also helping zoos here, trying to help with the enrichment program. So that 1 microliter is changing the world in some ways. Okay, so if then that microliter is not in my system or in my assay that I'm developing, I will not get the best or the appropriate results that I need. And this is the training that I'm giving to each and every student, that it is very important that you understand how an assay works. An enzyme immunoassay is basically antibody-antigen reaction. So it's actually in a 96-well plate in which you coat your antibodies and then add your antigens.
So these antibodies are produced by animals, and they bind to specific targets, which we're calling an antigen.
Yes.
And they're actually used by the immune system of animals. Yes. And you can give me a hug later, man. Yeah. They're actually used by the immune system of animals to kind of target specific things in bacteria or anything that's invading the body.
Yes.
But the way scientists use them, and the way that you would have used them to develop tests, is by making them bind to specific targets. So in this case, it's going to be like the cortisol of the frog.
Very good, yes.
So we have these antibodies already that target cortisol for humans and probably some other species.
Yeah.
So we can measure stress responses by taking the
Yeah.
blood. But you've developed an assay, one of these antibodies that binds to the cortisol in the frog. Specifically.
Yes.
And you can detect that in the frog's urine.
Yes, and that is amazing.
That's a good
I think I should get you as a PhD student right away. And you can see here, if you can show it closely.
You can show that, and you should flip to the 96 well plate a bit
as
off.
well. So that's basically the biological validation that I was talking about.
That's a spike post-induction of ACTH.
No, that is actually the HCG, human chorionigraphic gonadotropin for the testosterone.
Okay.
Okay, so we have got similar one for the ACTH. It might be.
Just, I guess a question in regards to developing these antibodies. Did you develop the antibodies yourself?
We did not personally develop them here. It was actually done at the UC Davis, California, by one of my colleagues.
And so they'll
So-
do that as they do with the development of any antibody. They would inject an animal with a certain antigen, which is the
Yes,
the
yeah,
cortisol that you're
yeah.
trying to test for, and
Yeah.
then collect
Yeah.
antibodies of that
Yeah.
animal. Okay.
I can't find that 96
Yeah.
well plate, but that's okay.
I'll show it to you. No, I'll show it to you. I'll show it to you. So now what we've got is actually a whole society of non-invasive biologists,
Yeah.
and that is known as the Society for Non-Invasive Endocrinology.
I guess it's important as well. If you're measuring stress in animals, if you're doing an invasive stress like trying to take blood, and as you say, it induces stress, that's going to actually probably interfere with what you're trying to measure as well. So it's not even probably better from an ethical perspective and not causing animals
Yes,
harm, but it's also better from a research
Yes.
perspective.
Because you can't tell if it's the data collection or the
Yeah.
actual stress.
So, for example, I have got a student who was in the lab for the past 2 days. She is looking at small mammals here in Australia, looking at antikinase and rat species, and trying to understand the landscape of fear. So that project actually will try to understand the devastation that humans are causing to the land. Does that actually increase the number of predators that we have in the system, or the accessibility of predators to small mammals? So that is a big project. Okay, and to be able to answer that, we need to do some pilot studies. So we are doing some pilot trials in a more controlled environment at a local zoo, where we are stimulating predatory cues to the actual small mammals and collecting their stress hormones, which is basically in scat samples. So these hormones are measurable. And like I said, we've got the Society for Non-Invasive Endocrinologists, of which I am a part. We actually supply antibodies to anyone in the world. So we've got clients from India, we've got clients from all over the world, zoos, and it actually is a one-stop shop rather than going all over the place, because I review a lot of papers as well in my area of expertise, and I can see that, for example, there was a cool research on Komodo dragon, and they came up with a technique to actually measure hormones in the sloughed skin of the dragon as an index of chronic stress. As you know, the distinction between acute stress and chronic stress, and we can't do it in blood, as you were right, that it is quite invasive, and it is episodic. So as soon as before this technique was available, I was basically jabbing frogs, cane toads with the needle, and you have to collect blood from the heart without puncturing the aorta. So that itself comes through a technique, and it's basically hands-on. You can't write it that you injected at that level, and this is how you'll. So it is very hard to teach the students as well. The questions are interesting. Everyone wants to study it. So, the Komodo dragon study was basically measuring these hormones in the skin, but their validation was basically very, very weak. So the editor-in-chief wrote back to me saying, Edward, I love this paper. The title is hot. I said, look, the title is hot, but the work is cold. Okay. So as a reviewer, I had to do justice, because what will happen is people will pick up this paper. For example, Edward studied the ground frogs, more than 10 years ago. His paper is being cited. I feel so happy, and that gives me pleasure as a researcher, that students, young scholars email me, from all over the world. I've got a student coming from the Zoological Society for London in the next few weeks. She's looking at rescued turtles, and we are trying to see how stress levels change with response to tendering love and care to the turtles that we provide. That work will be done in canes. So it gives me a bit of a pleasure that, that, like you talked about my journey, the steps that I've taken being a student, and I have that strong connection with my students. I understand the phases that they are going through, and only through experience. If I did not have that experience, if I wasn't put on the front line, the project in Fiji, like people were laughing at me at the time, saying, Edward, with Fiji's socioeconomic and political situation, you will never be able to do a project there. And never knowing that the frog has got hormones in its urine, coming to the airport and being stopped there, and then having only 3 months to finish and wrap up. So within my PhD, we were the first group to actually publish that we can measure hormones in frog's urine. So that was the novelty of my PhD. Like for every PhD student, there needs to be a novel component. So getting the eggs and doing the captive breeding, that was also novel, but this was it. And I never knew that I would be doing that. It was a blessing from somewhere, and my supervisors are still, email them and thank them at every stage, because whatever I do, I reflect back on that first day. Because I was a gold medalist in chemistry. So my opportunity when I was given the medal, Shell Fiji, which is a big fuel company, the director was there and said, Edward, I'll give you a job. I said, okay. And then I went to my mom, and I said, Mom, should I stick to chemistry or should I go to biology? Because biology is something is in my heart. I don't need to revise. I don't need to study to get, good grades in biology. And all the chemistry lecturers were like trying to hang me on the wall so that I don't run away, because they were looking at coming up with new chemicals and things like that, chemical engineering and all that. But one of the big chemistry professors at that time said, Edward, all these labs will close down in 10 years' time, so you might have no job. So, that
you
was...
also need to do what you're passionate in life.
And that was it. It was my mother, actually, who gave me that answer. So I was standing in the hallway of this university. The chemistry department is on the right. The biology department is on the left. And I'm standing there, and because I had got the PhD upgrade, and the chemistry professor was prepared to give me a PhD in chemistry, he had lured me in with he said, you can teach my lectures, I'll give you this much money. And I felt a bit scared as well as a student. What should I do now? So I called Mom and said, Mom, this is what's happening. Said, Edward, just go with your heart. Biology is your thing. So the chemistry professors became a bit unhappy. I just took my bag and said, I have to go now. But still, after I think 10 years, he's moved to Australia, and he gave me an email and said, Edward, I'm so proud of your achievements. And so these are little decisions that you make in life that makes a big difference. You don't need to, it happens, you don't need to think very hard about it. When I went into doing biology, it was something, it was my passion. And look at where I am now, coming to Australia and getting these opportunities, I'm blessed with it. Within that journey itself, when I came here, 10 years ago, I did my postdoc at Griffith University in Gold Coast. So for 4 years I was there, and then the Australian Academy of Science sent me to India to set up a program in conservation physiology in Maharashtra, which is like one of the hub for frogs and lizards and all these cool animals. We went, there's another big story, the monkey stole all my lollies and all that. So, that relationship is there. And then as part of my journey, I went to Canada as well, to Saskatchewan, did a third postdoc looking at goldfish as a model for metabolism. because this guy from Saskatchewan Uni had heard my talk in India, and he said, Edward, we should collaborate, because he was interested in metabolic genes. So they discovered this new gene that can actually regress your hunger. So he said, why don't you look at stress? So at that time, my postdoc was running out. So, we compete with a very little pool of money. And I basically packed my bag and left. All my friends were laughing at me. Said, Saskatchewan is like, it's very small and all that. I said, no, don't worry. Education takes you anywhere, wherever you have to go. I went there, in the snow and all that. Hadn't seen that before, coming from Fiji and all that.
Yeah.
That was an experience. So after that, then I got a lecturership. Because coming from PhD to doing a postdoc, getting a lecturership is very hard to get. It requires a lot of experience and a lot of publication. So that journey was important for me to have done these postdocs, these little stints here and there. And at one stage, it came that I had 3 offers sitting on my table, all with 6-figure salaries. I was sitting there and deciding which one to pick. Coming from Fiji, that journey, and now with 3 offers, like I was sharing this to my student just last week. I never knew that, and it comes all through. It will come, for students. You just have to believe in yourself. I was, from a place with very limited scientific support. Even I went back to Fiji and gave talks at all universities there, and they really admired and appreciated, and the professors there know that I'm in Australia and doing all this stuff. So it's very important to have, as a student, like the message.
Yeah.
It seems like the key to your success has been following your passions and working very hard and not giving up, and also taking risks.
Yes. I am very good at putting myself in the fire and coming out unscathed.
Do you have, what about...
Wait, I think that's something that students can take, but in regards to your chemistry and biology, look, I couldn't choose which way to go, because I was kind of like split, so I decided to do biochemistry.
That's even better.
We're kind of like...
Do you have, are you taking students at the moment for any projects? Do you want to spruik any projects?
We are about to start a big project on, again, this is looking at goldfish as a model for stress. Because what's happening is we are moving away from invasive methods. Next year, there will be an opportunity to actually collect water samples as an index of stress. So we are doing this pilot study. It will start in spring, where we are actually looking at gene expression. So it's a project entitled "Does the Body Talk with the Environment?" So we are measuring responses in goldfish, because it's a very good lab rat as such, and basically subjecting them to various physical stressors, mild ones, and then collecting their tissues, and then all the way through to the water in which it is sitting. And I think that will give a very 3D sort of a connection between the brain, the body, and the behavior. And next year, we are hoping to actually expand into blue mountain skink, reptiles, frogs, and look at across gradients, how they are actually responding without having to actually harass animals, collect blood and all these things. And thanks, and that can be a part of our ongoing conservation effort. Apart from that, I have a lot of interest in domesticated animals as well. So cats, dogs. We've got projects looking at sheep. Okay, so farm to the plate approach, looking at meat quality. So, that's another, I could sit here and talk for the whole day. But basically, we are trying to see whether we can minimize stress on farm animals so that the quality of the produce is maintained. Because if a meat, if you know that it's actually the glycogen bucket, so that glycogen bucket is buggered up if the animal is stressed. And that's $1 value lost for the producer. For every kilo, I think he loses 50 cents. So if you talk to any big sheep farmer in Australia, so we've done that research for over a few years now, that we found actually in wool samples that if the mother is hot, as in she has been sitting in the sun and she is facing climatic stress or heat waves, stress hormones are high, and we measured the stress hormones in wool and feces. So this is entirely non-invasive. And then she actually drops her baby quite early, and that is also lamb loss for the farmer. So we have got these projects, and then there's opportunities. It's more about what the student is interested in. So, for example, I've got a student who came to me last week and was interested in sun bear in Vietnam. In Cambodia or some other place. And so, like I said, it's not about, Edward, give me the results right now. It could be something else. For every project, we need to sit down and weigh out the pros and cons. If it's an international project, we need to look at it, whether it's feasible, what is possible, what is not. So, for example, with the bear study, we said we might just look at stereotypic behavior rather than collect scat samples, because I've already done the scat stuff. The stereotypic behavior, chronic stress, can it be reversed? That is a very interesting question. So the lab is growing. It's the stress lab, and we have got visitors coming from the UK and from different parts of the world giving talks to my students. And it's basically a student-driven lab. And if anyone is interested in even just seeking advice on career paths or something like that, I'm very open and able to help students. Thank you so much.
Thanks a lot.
Cheers.
Thank you.